Hello and welcome to Biology Explained. Today we're going to be looking at the Hardy Weinberg principle as part of my A-level biology revision series and how we can use these calculations and assumptions to work out if any kind of gene flow is changing from one generation to the next. So let's first talk about what we're kind of meaning by population here, which is integral to understanding what we're looking at with this Hardy Weinberg calculation. So population is a group of organisms of the same species living in a particular area at a particular time, and they can also interbreed with each other. As we should probably know by now, a species is defined as a group of similar organisms that can reproduce to give fertile offspring. Now, species can exist as one or more populations, but for this example, we're just going to discuss one population. Now, within these populations, we have what we call a gene pool, which is a complete range of alleles, and we're saying alleles is kind of different variants of a gene. So one gene could have 2, 3, 4, 5, and upwards, different types of alleles that do slightly different, make the gene do slightly different things when it's present in an organism. And this gene pool contains all the different types of alleles for all the different genes in these organisms in this population. And so different populations can have different gene pools made up of a different range of alleles. How often a particular allele occurs in a population is called the allele frequency. And in this case, it's usually given as a percentage of the total population, such as like 55% or 0.45, 0.55 as a number. So we need to know if evolution is happening. That's what we want to look at here. We want to know if evolution is occurring, if specific alleles are taking precedence over other alleles in a population as the generations go on. And so you want to be able to see if a allele is kind of becoming more and more popular as more and more generations of this population continue over time. And so what Hardy Weinberg did when they created these kind of calculations and this principle is that they created a null hypothesis. So they wanted to show, using a calculation, to show that if no evolution was happening, allele frequencies wouldn't change. And so they wanted to create a calculation to do this. So they would predict the frequencies of alleles in a population don't change from one generation to the next. And then they will kind of do a calculation, which we'll go over later, and then decide whether or not evolution is actually happening from this. However, what they're assuming here is that there are certain things that are, happening that they have to assume are happening in order for the model to kind of work. Otherwise, it gets massively complicated real fast, and you'll have to use much more advanced calculations. So it keeps it pretty simple. And so what they're assuming here is that there is a large population, so it's a representative population, and there's a lot of gene flow going on between the different animals. There's also no immigration or emigration, so no genes are coming in or out. There's no gene flow in or out of the population. There's no random mutations, so there's no new alleles appearing which may be more popular than alleles currently in the population. And there's no natural selection, so no alleles are being kind of picked for because of the environment these species are in. So an allele which, makes this bear immune to fire or whatever is not being selected for because the bear lives in a volcano, essentially. So that's not happening. And they also have to assume that there has to be random mating. So, some alleles don't provide, sexual benefits or whatever that improve their chances of mating. All the animals can mate randomly with each other, and then there's no selection because of that. So this means all the possible genotypes in the population can breed with each other equally. You can then use the Hardy Weinberg equations, which we'll talk about in a bit, to calculate the frequency of particular alleles, genotypes, and phenotypes. So it's 3 different things within a population. You can then also use the calculations to test whether or not the Hardy Weinberg principle applies to particular alleles in particular populations. So you can see whether selection or any other factors are influencing allele frequencies. If the frequencies do change between generations in a large population, then there is influence of some kind, as I mentioned earlier. But we'll get to that at the end. First, we have to understand how these calculations in their basic use can be done to kind of just calculate the allele frequency, the genotype frequency, and phenotype frequency to begin with in this population where there's all these assumptions occurring. So it's like fairly stable. So if we look at the first equation, and this is used to predict allele frequency. So as we were saying earlier, alleles are different variants of one particular gene. So you can have, like, let's say there's only 2 alleles of this particular gene. One allele is dominant and one is recessive. So the dominant one will probably have a higher frequency in the population because it's more likely to be, because it's technically more dominant, so it's more likely to make its way into the population. You'd assume, although that's not always the case. So that's what we're looking at here. We're seeing which one is more popular. Maybe all these recessive alleles have been passed on for some reason. So this first calculation is pretty simple. It's just P plus Q equals 1. And we'll talk about what that is. So we're assuming here, and it's generally assumed for a Hardy Weinberg equation, that P is the frequency of one of these alleles out of the 2 I was talking about. We would classify P normally as a dominant one. And then Q is the frequency of the other allele of the 2 alleles that we have in the population. And that's usually the recessive one. Now, the important thing to note here is that all the alleles we could add together, in this case P and Q, but if there was more alleles, you could add more, P plus Q plus A plus B, so you could have 4 alleles added together. Whatever the case, the frequency of all the alleles in a population always equals 1. So let's say all the alleles for a particular characteristic always equals 1. So P plus Q will always equal 1, or however many alleles there are for a particular gene. So if we took, for an example, a species of plant with red or white flowers, where red, big R, is dominant, and white, little r, is recessive, if the frequency of red, big R, is 0.4, then the frequency of white, little r, is 1 minus 0.4 equals 6. So you can just simply rearrange this calculation. So if you didn't know P, the P value, but you knew the Q value, you'd move the Q over the equal sign. So you'd minus it from both sides and then have 1 minus Q, so 1 minus whatever that number is, and that would equal P. So it's a fairly simple calculation to work out how the allele frequency of all the alleles for a specific characteristic are. So as long as you remember they always equal 1, it's a simple rearrangement calculation from the information given to you. So that's allele frequency. So that's important to note that, as I've mentioned before, and I'll mention again and again, allele frequency is, alleles are, the different types of one particular gene. So each gene can have many different alleles, or one allele. It varies depending on each gene. So if we look at this table, we can see that for, let's say, a human, each gene you have two alleles basically on your chromosome. And so in this case, these 2 people, one has big A, little a, and the other has big A, little a. And we're classifying big A as the dominant one and little a as a recessive one. And so that's what we're looking at there. And in this case, because this is the genetic cross we're looking at roughly here, it's a Punnett square, as you've probably looked at earlier. We're saying they're kind of, haploid cells, so each gamete only has one allele in it. And then they're being crossed over to create a possible chance of creating a diploid genetic cross. So we're looking at the alleles, the single ones, and then they're being combined together to create a genotype, basically. So if we go on to next, we've done that first calculation, which is looking at allele frequency. If we then go and look at the next calculation, we're going to look at how to predict genotype and phenotype frequency. So a genotype, as you should know, is an organism's set of genes that it has been passed down from its parents, while the phenotype is kind of the way the genotype is presented to the outside world. So the genotype will be big A, or big A, or little a, but the phenotype will be, big A is classified as a red flower, or little a is classified as a blue flower. Phenotype is what it looks like to the outside world, while genotype is what it is genetically. So once we know that, we can then try and work out the frequency, because the genotype frequency and phenotype frequency are not always the same. And I'll discuss that in a second. First, we have to look at how we kind of get the calculation that allows us to work out genotype and phenotype frequency in a population. So you can look at this Punnett square, and that helps you work it out. So if we have the two dominant genes being crossed, we get p squared, because when you go from alleles to a genotype, you always have two alleles in a particular genotype. Yeah. So if we then do this cross, we can see we always have a p squared one, two pqs, and a q squared one. And that gives us a calculation because this will always be the same. So let's say p squared, right, is the homozygous dominant genotype frequency if p is the dominant allele. And so this is just how these calculations always work out. So as long as you remember, p squared is the frequency of homozygous dominant genotype. So by homozygous dominant, we mean two of the same dominant alleles become the genotype. And then for 2pq, it's the frequency of the heterozygous genotype. And the reason you have two is because when you do that basic Punnett square, 50% of them are the heterozygous genotype. And then for the same for q squared, for the recessive genotype, that's homozygous because 25% of this cross will be the homozygous recessive genotype. So if you think about it, it's the same as a normal Punnett square, just you're writing it as letters instead of, the genotype as you would in a normal Punnett square. And by writing it as those letters, it allows you to write it out in this calculation, p squared plus 2pq plus q squared equals 1. And the reason it always equals 1 is because the frequency of all possible genotypes in a population is always equals 1. So similar to how all the allele frequencies in a population equals 1, all the possible genotype frequencies in a population equals 1. So if we just think about it, so if we were to say for a basic calculation, if there were two flower colors again, we have P and we have Q, as you can see on this Punnett square. And then there are three possible genotypes, P squared, PQ, and Q squared. So if we were to say the frequency of genotype P squared, or AA, is 0.34, and the frequency of genotype PQ, or big A little a, is 0.27, the frequency of the genotype AA, little a, or Q squared must be 1 minus 0.34 minus 0.27 equals 0.39. That's the frequency of Q squared. So again, you've just got to rearrange this equation to get what you want. But you have to remember that if there's only three possible genotypes, the number it gives you for the frequency of genotype for the heterozygous genotype will be, for these doubled one. So we were saying that the frequency of the genotype big A little a, or PQ, was 0.27. That in this calculation is the 0.27 is equal to 2PQ. So you don't have to worry about that. But this is where it gets a little bit more confusing, and you've got to remember is that phenotype frequency does not always equal genotype frequency. But you can use the genotype frequency to work out phenotype frequency. So if we were going to say the frequency of red flowers is equal to the genotype frequencies of Rp squared and 2pq added together, so that's 0.34 plus 0.27 equals 0.61, and then the frequency of white flowers is equal to the genotype frequency of 0.39, and that equals 1. So in this case, there's only two possible phenotypic outcomes. So if it was to say all those red flowers equal 0.61, the 0.61 encompasses p squared plus 2pq, because the phenotype is only looking at what is expressed. So p squared is the dominant one, so the dominant will be expressed, and 2pq, because it's heterozygous, the dominant color will be expressed, so red again. While the recessive one, q squared, will be, that will be expressed, but that will probably be a lower number. So you've got to make sure to look out for, if it's asking for phenotype, you just got to make sure, you're including the correct bits of the equation into a specific thing. Whereas genotype will be, each part of this equation will be a specific number. Phenotype could be bunching, part of the equation together. So just watch out for that. And comment if you're unsure. So now we can look at an example calculation to try and help you understand what is going on here. And so this is an example A-level question, which is worth 3 marks. So they've given you the equation, or one of the equations, although you should be able to remember the other equation if you need it, because sometimes you need both of them. So the Hardy Weinberg equation can be used to estimate the frequency of a recessive allele in a population. Hemochromatosis is a condition caused by a recessive allele. In one country, 1 in every 400 people, so you should highlight that because that's a percentage of people that they've given you, people were found to have hemochromatosis. So the question says, describe, so you need to just basically write out the calculations. So so you got to remember, describe and explain, maybe ask for different things sometimes. How you would use the Hardy Weinberg equation to calculate the frequency of people who are healthy but carriers, so heterozygotes of the allele for hemochromatosis. So you've got 2 things here. So you've got a number, 1 in every 400 people have hemochromatosis. Describe, should highlight, and to calculate the frequency of people who are healthy but carriers, so heterozygotes of the allele. So this is important to know, because the bit you're looking out for is the bit that's important to know here is it's asking for the allele frequency. So you're going to have to use p plus q equals 1 at some point in this calculation, and that's where people can get tripped over. Yes, you're going to have to use p squared plus 2pq plus q squared equals 1 to begin with, but then that only gives you the genotype frequency. You're then going to have to use the calculation p plus q equals 1 to work out the allele frequency at some point in this calculation. So what we know is that 1 in every 400 people has hemochromatosis, which means because hematosis is a recessive allele, that will be our homozygous recessive genotype, which we're calling q squared. So they've given us the number for q squared, basically. And therefore, we want to do alleles here. So this is an important thing. The bit not to get caught out with is they've given you p squared plus 2pq plus q squared equals 1, but you don't really need to use that calculation for this particular question. Because it's asking for allele frequency, we just need to get the allele. So we know that the q squared number, so that's the recessive genotype for this calculation, is 1 in 400. So all we have to do is square root that calculation, that q squared. So we need to square root 1 in every 400 to get our recessive allele frequency instead of our recessive genotype frequency, if that's clear. And so when we do that, we get 0.05 is the frequency of the recessive allele. And then, because we now have the allele, we have q, we have the recessive allele q, we can put that into our p plus q equals 1 calculation, rearrange it, and then we get our dominant allele frequency in the population, which is 0.95. So we now know the recessive allele frequency and the dominant allele frequency in the population. So that's great to know, but then we have to read the question again. So we're going to be looking at, we want to know the frequency of people who are healthy but carriers, so that means they're heterozygous, of the allele for hemochromatosis. So we've now, because we've already worked out the frequency of each individual allele in the population, all we need to do now to find out the frequency of the heterozygote genotype this time is scale it, change it back into the genotype from the allele frequencies. So we go back to our first calculation, the p squared plus 2pq plus q squared equals 1, which is our genotype calculation or phenotype calculation, and put in our allele numbers we just worked out. So we do 2, and for the p we do 0.95, and for the q we do 0.05. And therefore our answer is 0.095, and that gives us the frequency of the heterozygous genotype. So it doesn't seem too bad as long as you remember sometimes you've got to change it from genotypes into alleles and then change those alleles back into a genotype. So To do great in these questions, A, requires quite a bit of just practicing them and get work, not being caught out by doing past paper questions on it and making sure you understand what it's asking you to do. Not being caught up on the difference between genotype, phenotype, and allele frequencies. And also just making sure to read the question so you don't get carried away and write genotype frequency when you meant to write allele frequency. Any questions, feel free to comment, but please like and subscribe if you found this video helpful. Thank you. Stay tuned for more.